welcome to VIVA Unscripted. This is sort of the conversation after the conversation. And I have with me Dr. Aloke Finn on my left and Dr. Marc Schermerhorn on my right, who are two of the late-breaking clinical trial presenters. Welcome, guys.
Thank you.
Thank you.
So obviously you both did an exceptional job up there, and our panel did equally well in kind of addressing some of the questions. But this is our opportunity to sort of be a little bit more informal, kind of dig into this a little bit more, and perhaps from my perspective, more importantly, kind of get your insights beyond the data. Mm-hmm. So when I start with you, Aloke, obviously this whole idea of downstream effects of drug-coated balloons has been a hot topic. Industry has used this or weaponized this, in some respect against each other. How really clinically relevant is this? I mean, did what you see in swine really translate, in your opinion, to clinically relevant downstream effects that might, affect wound healing, for instance?
It's an excellent question. I think it's difficult to know the exact relevance in humans, especially because a lot of the patients selected for trials are highly selected. The ones that have critical limb ischemia, that have really bad downstream distal disease, impaired healing, for instance, are the ones where I think these emboli would be most impactful. And it's very difficult to know what the consequences are. Although it must be said that because we understand what paclitaxel can do, and it basically causes tissue death and necrosis, it's important that we minimize emboli, even if we don't really fully understand their clinical significance. And I think balloon technologies really need to aim at being able to treat the target site while really minimizing that downstream embolic effect.
Right. I remember some years ago I chaired a CEC on a paclitaxel wrap that was put in the time of a surgical hemodialysis fistula, and that trial stopped early because the wounds didn't heal.
That's right.
So you think this is a real threat? Is it the lower drug? Is it the excipient? Is it the dwell time? What are the other factors that might impact this?
I think part of it is the excipient, part of it is the crystallinity of the paclitaxel. The particles themselves differ between balloons. So I think part of it, what we're seeing is that the amount of emboli differs certainly between balloons, but also the size of the particulates that are released can differ. And the size may determine where those particulates lodge. Some may be passed through, but some may lodge in arteriolar beds, causing tissue ischemia and necrosis, and I think that's the real concern. So understanding more the size of the particulates that are lodging and their significance, I think, is a very important thing we're going to have to explore in terms of the DCB technologies.
Marc, sort of switch over to some of that engaged data. Really interesting. We saw in your presentation, as well as Gary Ansell's presentation on carotid, is that these gender differences seem to be kind of minimized as we move forward with technology development. What do you think this is? What's changed in how we deliver therapy or understanding these treatments?
Yeah, I think there's probably several things at play. I think primarily it's advance in the technology. I think with lower profile devices that are more flexible, better hydrophilic coating, we can get through the more challenging access anatomy for women. And I think with the ability to do a slow, careful, more accurate deployment, you can deal with a shorter proximal neck. So I think those are probably big advances in terms of allowing us to treat women better. And part of it is, we're becoming more experienced, and maybe we're selecting patients better. So I think it's probably a combination of things. But I think hopefully we are starting to bridge that gender gap and at least now have equivalent outcomes in the patients that we do select, because that's something that we didn't see routinely in the past. And hopefully, as this technology continues to evolve, we can actually get more and more women to be eligible for an endovascular approach.
Especially with aneurysm disease, where these are large bore devices, and the perceived differences in anatomy are scary, right? I mean, you're worried about a greater risk of rupture in delivering these things,
Sure.
causing real dissection or vessel injury. Do you think that women are underrepresented in aneurysm trials and in trials in general? Do we need to assure that there's proportional enrollment of women in future studies?
Yeah, I think we probably should go out of our way to make sure there's enough representation, because we're using these data to make broad generalizations to the population. And I think one other thing we should probably look at maybe is the proportion of patients that got screened and didn't make it into the trial, just so we can know better, how generalizable these data are and how many people are going to fit the various IFU.
So you're given here a blank check. It's not going to happen, by the way. But were you to be given a blank check, what would you like to see sort of moving forward in terms of future aortic registries and/or future trials? I mean, what are some of the outstanding questions that you can, now identify based upon ENGAGE and obviously your other experience?
Yeah, well, I think more registries like this, longer follow-up, because I think one thing we saw with the Medicare data is that you really need to follow these patients a long time, and then more and more adverse events start showing up. So I do think that very long-term follow-up is important. I think extending it out to 10 years for this study is great, but I think it'd be best if we could enroll every patient into a registry, whether it's better clinical data from Medicare or other payers, or if it's getting more people involved in VQI. I think that's a great way to gather more data, and then also see how different grafts compare, and hopefully that'll stimulate manufacturers to make continuous improvements, improving profile, deliverability, et cetera.
I've always thought that we could learn from the orphan drug industry and have smaller clinical trials and larger post-market surveillance, with the understanding that, that post-market surveillance does provide an opportunity to, evaluate and discontinue commercial availability of devices, but certainly get people access earlier and get kind of, that real-world data.
Yeah, I think that's a great idea, and I think, some manufacturers are doing it, and I think that's great, and I think we as surgeons and interventionalists should participate in these registries and enroll as many patients as we can, so we do have a way to track their data.
Who's going to pay for those registries? Is it incumbent upon physicians? It should be a mandate of institutions as performance and quality improvement that they collect and report data? Yeah,
I think the institutions should pay. I think it's not really appropriate to force the physicians to pay.
Okay, I don't know about your place, but they want to pay me less. So let me ask you something, Dr. Finn. Is your model a reasonably robust model to interpret differences in balloon effects that we can apply this before these things become utilized in humans?
I do think the model is reproducible, and the reason I say that is because we actually used the same model in a previous publication using impact, and we also compared it to Lutonix. The rate of emboli and impact were almost identical to the rate seen in the study I presented today. So I think there is a lot of reproducibility in the model, and I do believe that what we showed today was representative of what really happens in vivo in patients with these balloons in terms of particulate emboli.
Now, this is obviously not an atherosclerotic model.
No.
Healthy vessels.
Right.
Would there be any advantage, or would it potentially change that? And what I think of is, we do, a lot of tibial intervention, and now that, whatever is on that balloon, even its protectant, is being scraped off the balloon as you go through this, atherosclerotic milieu. So, any way to sort of expand this model to better represent drug delivery in humans?
It's a good question. I think we hesitate to use atherosclerotic models sometimes in these types of studies because they add one more level of complexity, and they add one more issue that has to be accounted for in terms of comparing technologies. So, the atherosclerosis is the cholesterol levels aren't always even, the extent of disease is not always even. You have to account for that when you're comparing these different groups, and it's not always as clear cut as when you use normal animals. So, although I am a fan of using atherosclerotic models, I don't know if it's possible to interpret them as simply as it is in normal animals, because of the added complexity of the atherosclerosis itself.
Interesting. So VIVA is our opportunity to sort of showcase new technology and, kind of new advances. So what's on the horizon from the aortic perspective? I mean, what are you going to have to show us next year? What can we look forward to?
I think, the future will hopefully be further advances in, profile delivery, et cetera. But I think it's going to be, moving up and down the aorta. So, iliac branches, visceral branches, thoracic branches. I think that's sort of the next step that we'll start hearing a lot more about at this meeting and others.
Right. And, maybe you're better positioned to sort of look at this, but, if I think of this meeting 10 years from now, I think that it's going to be a meeting where we see a lot more biologic and polypharmacy discussion, and how this is married, both relationship to devices and independent devices. I mean, what's your kind of future look?
I think we spent a lot of time focusing on paclitaxel as a drug because for DCBs, because of the tissue binding characteristics, it's easy to transfer that drug. It's much easier to get drug effect without having sustained release. But remember, paclitaxel's therapeutic window is fairly small. The downstream emboli can be toxic. Will we eventually move towards sirolimus or mTOR inhibitor-like molecules in the future? We currently are limited because they don't really have the same tissue binding characteristics as paclitaxel. But as we improve our drug transfer technology, I think we may move more towards those type of agents rather than paclitaxel, simply because they're better tolerated.
Right, and I don't know if you saw the disruptive tech, but we talked about some new systems, isolated perfusion systems. Some years ago, I worked with Abraxane on nanoparticle paclitaxel.
Yes, that's right.
Which had sort of better binding affinity.
That's right.
So I think you're absolutely right. The idea that different agents, different delivery models, and maybe more than one agent.
Right, exactly. And are we going to differ our treatments above the knee and below the knee? Are we going to subspecialize our technology so that we have different selected technologies for different indications?
Right. I mean, I find it extremely arrogant of us as physicians to think that our $100 million year old evolutionary body can be impacted by one drug,
right?
And one delivery model.
Absolutely.
And clearly, our biological redundancy is much more complex.
Absolutely. We're in the I think we could look at it as sort of a simplistic stage, and we will advance as we go, just as coronary technology has advanced.
Right. Let me ask a question: baseball fan or football fan?
More of a football fan for sure, but that's kind of decreasing recently.
Right. NFL football or
NFL.
Soccer?
NFL. Right. Oh, soccer much more than NFL.
Well, I don't know anything about soccer.
Okay.
So, what's your pick for the NFL?
That's a great question. I don't have a good pick. I think that I'm from Washington, D.C., so I was always a fan of the Redskins.
Right.
But and they were very good when I grew up, but they haven't done so well recently. So I don't have a great feeling about them this year.
Cousins is a solid fantasy player. Right? What do you think? What do you think about the Major League Baseball this year? Dodgers had a good run. It's kind of died down here. What's going to happen?
Well, I'm from Boston, so
I'm sorry to hear that.
Go Sox. We're hanging in there right now. My Patriots, which is my primary team, had a bad day. Yeah. But I'm hoping they'll be able to turn it around.
You guys have too many new receiver targets. It's you got to kind of pull that cord together.
But the other football is my favorite too. So Barcelona, did well against Juventus in the Champions League, and I think they got a shot at.
This is where I leave the dialogue. You guys can talk amongst yourselves. But I want to thank you both, obviously, for your great efforts here.
Thank you.
Appreciate the quality of those trials and for participating in this. So
great. Thank you very much.
Part of our faculty.
Appreciate it.
Thank you.